Welcome to BuzzLocal.tv. We're taking it up a notch on the intellect level. We have Andy Roberts here, who is the co-founder of Teos Pharmaceuticals. Now that is a name that kind of leaves to the imagination. So fill us in. What exactly is Teos Pharmaceuticals?
Exactly. So Teos Pharmaceuticals, hopefully in time not too distant from now, will have many things to offer patients that haven't previously been offered, but certainly our first project relates to cancer, the big bad disease of cancer. So most people know or have been affected by cancer directly. 1 in 2 males will be diagnosed. 1 in 3 females will be diagnosed with cancer throughout their lifetime. And the issue with cancer is one that our results have been less than stellar. So if we look back in the last 40, even 50 years, back to Nixon's time as president, and we adjust mortality rates for age, we actually see a trend across the board where things haven't gotten a heck of a lot better. So anytime in science, and as a scientist, when you see things not improving for the better, especially when patient outcomes are your measures, you really want to sit back and reassess exactly what do we know about this disease, what underpins it, and are we effectively targeting that underpinning cause. And if we can't boast tremendous results, then we have to take that step back and really have a hard look at this. So we've seen this many times in medicine. You know, crazy things like washing your hands made a huge difference. We had people with fevers who didn't want to eat, who were sweating through their bed sheets, who had low blood pressure, and all these symptoms, and we kind of figured out these were all underpinned by a bacterial infection. So of course the onset of antibiotics made a huge difference in that. And boom, all of a sudden you went from having horrific numbers to exceedingly good numbers and very curable rates. We've seen that in HIV, where the single drug approach just didn't seem to work great. And all of a sudden we used drugs from similar classes, antiretrovirals, and in combinational therapy we've basically in a generation taken HIV from a virus people were exceedingly scared of, to a virus that we expect patients to live a relatively normal and healthy-ish life, so to speak. So with cancer, kind of in that same light, our company Teos, which is 4 MDs, and all of us are practicing clinicians, meaning we're not full-time researchers, we've been compelled by an emerging, or I should say re-emerging body of literature that looks more closely at some of the patterns that cancer spells out for us. If you've been following cancer research for the past 40 years, we've kind of been resting on the assumption that these cancers are being driven by underlying somatic mutations, so mutations in our nuclear matter. And we've derived numerous therapies that target these specific mutations, and again, the results have been lackluster at best. So we wonder if this might be another case of us looking somewhat at the symptoms and finding some treatments that go after these symptoms and extend life to some degree, but really without affecting mortality benefit. We wonder if there might be something more to that. So Teos is a company whereby we're using world-first artificial intelligence, algorithm learning, super Google computers to map out the metabolic pathways of cancer and correspondingly develop custom-made compounds to attack those metabolic pathways. And it's interesting because I would expect most viewers don't have much experience in pharmaceutical development, but one of the older wings of pharmaceutical development is in an area of drugs called small molecule inhibitors. Mm-hmm. So in diseases, we like to draw out the pathways where a cell goes from normal to abnormal, and we look at those various steps. So at each step towards normal to not normal, there's an opportunity to intervene, there's an opportunity to inhibit, so to speak. And if we can develop a compound that effectively targets that space, then feasibly we can stop that normal state from transcending down to an abnormal state. So those are called small molecule inhibitors. The problem is we have lots of interactions we don't desire. So with small molecule inhibitors, we hit the target quite often, but as often we hit spots that are not intended to be targeted. So we call these off-target effects or side effects. So this is why you look on the back of any prescription or over-the-counter pill you take from Shoppers Drug Mart, and there's going to be a litany of conditions that may result from the taking of this medication. Those are off-target hits. So what we've been able to do with this artificial intelligence platform is map out every protein in the human body in 3D and then create compounds that hit the targets we want, and then we can simulate and visualize these off-target effects. So the idea was, rather than having a pill or a compound that hits target and then off target, why not hit when it hits target and then a bunch of additional targets? So create synergy. And kind of the visual way I might explain that is, if this is the receptor or the target I want to get after, then the molecule ought to look something like this. But oftentimes it ends up looking like this. So we get the interaction we desire. But the problem is, given this little three-dimensional change here, it might interlock with another protein ligand interaction like this. So these are the side effects we don't want. So if you're taking ibuprofen long term, it's great for anti-inflammatory, but it's not great in terms of developing a gastric ulcer. It's not great in terms of causing increased bruising because it interacts with platelets. So it's these off-target effects. So wouldn't it be neat if all of a sudden we designed a molecule that had this effect? So we have something that interacts with our target, but now we have a whole new structure that interacts with this and this and this, and these are additional synergistic effects. So what again, your viewers wouldn't notice this, but there's been a strong steer away from small molecule inhibitors towards very expensive, possibly inaccessible, possibly less efficacious therapies like immunotherapy. We were designed very specific antibodies and let them loose in a person's physiology. So the benefit is they hit the target you want. The downside is they don't hit anything else. So we kind of see the perceived disadvantage of small molecule inhibitors as an actual advantage when we can intelligently design these up front before testing them in animals, before testing them in humans. So we think with cancer, and from what we can decipher, we think we can do a much better job in terms of having efficacious compounds without the toxicity profile that cancer is so well known. I mean, cancer is essentially one of the only diseases out there where rational people, under the guidance of rational physicians, will opt out of therapy given the fact that the known side effects are so awful. And sometimes they can even hasten the death of the patient, and they know that very well. And we have these conversations with patients. So wouldn't it be nice if we could change things such that we have additional effects rather than side effects?
So basically, it's a new form of cancer drug, but minus this it's having the negative side effects being a negative, the side effects would be more of a positive. So we
can simulate all these things up front, and it's a lot quicker to do it this way, and it's a lot less expensive. So just to make you aware, in terms of developing a compound from concept to getting into a patient's mouth after clinical trials, the average cost is about $800 million.
Wow.
So that's in the range of kind of running for president. I mean, it's an exceedingly exclusive club in terms of who's going to have the ability to do that. So with novel technological support like we have access to, even lowly, community anesthesiologists have a shot at making real impacts on patient outcomes based on what we see clinically. So beyond the fact that we're taking a completely novel approach to the compound development, the underlying theory for which we're working is somewhat completely novel. So we've taken an older theory and we've kind of greatly added on to it.
Okay, sorry, like you'd mentioned that before, that this was an older theory that people had sort of gone past.
Yes.
And what made you come back to this?
So if you're a science geek, you know that anything worth reading in the science world before the Second World War came out of Germany. So Germany was the hub of science. This is where Albert Einstein was, amongst others. However, the most famous and most highly coveted cell biologist of all time was a guy named Otto Warburg. And research was interesting back then from the standpoint that these guys were put on very generous government stipends for life, with really the ability to do research in any area they wanted as they wanted to do it. So it was quite an interesting time for science. And he won the Nobel Prize in 1932 for an effect that's actually been named after him called the Warburg Effect. And what he noticed is, regardless of the tissue of origin that the cancer arose from, it had similar metabolism throughout. So whether it came from a prostate or a breast or a brain or pancreatic tissue, that the metabolic patterns were actually quite similar. So much so that we've exploited this Warburg effect in one of the most common cancer tests we have. So God forbid, if you got diagnosed with cancer today, we would want to see where it is. because where it is and where it isn't will change exactly how we provide you therapy effectively. So we'll provide you with something called a PET scan, and most people have probably heard of a PET scan, but almost nobody probably knows why it works. So with a PET scan, we bring you into the hospital, and I would inject you with a radioactive dye, and we would take pictures of where that dye is going. And that dye, lo and behold, would preferentially go to your cancer. So if your cancer was prostate or liver or lung, it would go there preferentially, and the uptake of the dye would be visualized through this radiological exam. So that begs the question, what quality does this dye specifically have that allows it to be uptaken by the cancer preferentially? And it's an interesting answer because what we use is glucose. So glucose kind of is composed of the three most common elements the universe has ever made: carbon, hydrogen, and oxygen. And cells love it, but cancer cells really love it. And we've come to find out that most of them are completely reliant upon it. They use glucose preferentially to drive the machinery of their proliferation. So there's more beyond this theory, and we probably won't have a lot of time to go into it. But that's our understanding, is that the metabolic alterations drive epigenetic somatic mutations. So these are the mutations I think we've been chasing for decades and decades, which in the end produce similar metabolic phenotypes, or another way to say that is metabolic flavors. So regardless of the patient, regardless of the tissue origin, these things run the same program every day. So the way I look at these is if you're playing defense on a football team, and the offense lines up a little bit differently every time, depending if they're from the prostate team or the breast team or whatever, but they run the same play. And we keep getting fooled by the formation they're throwing at us, not recognizing it's going to be a long bomb in the back of the field. So if we start looking at the patterns and the themes of the metabolism of the cancer, instead of looking at the mutations, then I think we'll be much further along, much quicker. In science, the quality of a theory it's in its reliability and validity. So can other people do it? Can it be repeated in other forms? Can it be looking from different angles and still be shown to be true? And is it true at all? So for example, if we believe in the theory of gravity, which is only a theory at the end of the day, if I let this go, and I won't, it'll drop, okay? But if I switch it up and use my right hand, and I hold it up and let it go, and it hits the ceiling, it's not a great theory. So the problem with somatic mutation theory of cancer, if we believe that mutations are entirely responsible for what we're seeing outwardly in terms of the cancer patient, then it's difficult to get your head around how some cancers, in fact, don't have any mutations. And then if we were to line up 5-10 patients with glioblastoma multiforme, a horrendous brain cancer, 5 of them would have completely exclusive mutations that we wouldn't see in the other 5 patients. If we took the average tumor out of a lung, a non-small cell lung carcinoma, we would expect, if we divided down the cells and looked for these mutations, we would find 60,000 or more different mutations. So again, if the theory's not strong through viewings of different lenses, then I think we're wise to look. And at the end of the day, is it valid? Because how have our numbers changed? How effective has this mode of therapy been? And again, there's lots of room for improvement in cancer. So we certainly feel that we've got a novel view of cancer, being not cancer doctors first off. And we certainly are using a world-first novel approach to finding compounds in this space.
From what point, like say from this point to saying, okay, we've got something tangible that we could actually try, for all intents and purposes, the numbers work.
So
how long will that take?
If I'm working for a pharmaceutical top 10 company, that average timeline is 4.5 years. So I would typically have to run about 10,000 to 1 million bioassays to have enough drug candidates that pass cell line testing, that pass animal testing to get to a clinical trial phase. Again, $800 million. With the use of this technology, we take that 4.5-year timeline of research and development with endless man hours, lab space, millions and millions of dollars, and we're going to do it in 100 days.
100 days.
So it speeds things up, and we're kind of on phase 2 of 6 at the present time. So we're still early days, but the feedback is very good. The simulations are going well. The compounds are coming together. So again, the pieces of the puzzle start to fit.
When you say this is running, is it like a computer program in very simple terms? In the sense of what you're developing, you're working to see if you can do this.
So these are self-learning algorithms. So so the more work we do with the system and the more questions we ask it, the smarter it gets.
Okay.
So it's interesting from that standpoint, but of course these computers can conduct operations, make observations, correlations, and view causality in ways that no human could ever expect it to do so. So with this program, it starts to become exciting in about 40 days from now, when things start to crystallize and we start making smaller and smaller and smaller modifications to ensure that more and more and more of our off-target effects are actually synergistic additional effects.
Is there ever going to be a point where it just kind of hands off and say, figure it out yourself?
Well,
that's kind of spooky. I mean, it's very exciting, but it's kind of spooky at the same time.
Yeah. So yeah, we're early days in terms of our project here, and so is artificial intelligence. To be truthful, this software has been highly validated in a number of ways, and certainly we feel as though it's the future of all pharmaceutical development. And we were lucky enough to kind of be at the station the day the train pulled in with an exciting project with a completely novel view of a disease that we see every day in our clinical practice. So yeah, at some point it does actually do that, and it comes back with completely interesting results. So for example, one of the things that we do is run the entire catalog of every compound ever used in this space. And the neat thing is the computer spits back, of course, not only the known effects that the patent holder is fully aware of and patented their compound with, it also spits out all these other potential interactions, which we're quite sure the patent holder has no idea there is. So this, there's many lines of thinking along with technology like this, whether you can repurpose already approved medications to better, treat patients in a more accessible, more cost-effective manner. So there's room for that also. We think, particularly in our space, given our novel view of what's underpinning the disease, that creating completely novel classes of compounds, and we think it'll end up being 4 or 5 classes at the end of the day, is probably going to be the best approach. We kind of view it similar to other diseases in that cancer is an especially unpredictable and smart disorder, in that it's going to be more than one compound. We would think in most cases it could be 4, it could be 5, it could be 8. But again, that would be exceedingly impossible to do on the present drug development paradigm. There's not enough money in the world. There's no company that would invest in this. It just wouldn't happen unless this technology was available.
That's huge. I mean, huge leap forward in a lot of things. I mean, just that. Once that's kind of done, I'm sure that same sort of technology then can be shifted to other issues and problems.
So there's 4 of us involved in the project. There's myself. The other co-founder is actually an anesthesiologist also, and we've hired him, and he's actually going to be coming here to Moncton next November to join us. And he and I have similar interests, and then we hired a couple of our friends, and they're no pushovers. So one's a Rhodes Scholar, finishing his MD PhD at Oxford right now. The other one's a triple board certified internal medicine, emergency medicine, intensive care unit physician at the Henry Ford Center in Detroit, who's also Mayo Clinic's 2018 pick for aerospace medicine at NASA.
Wow!
So there's some
no slouches there.
No, there's some good brain power with interesting conversations ongoing here for us. And then beyond that, we have 3 PhDs hired and working on this in Toronto with their novel drug discovery platform.
Well, that's awesome. I think probably maximum 12 words is what I took in and understood, but I think it was a good 12 words. I think it was the right 12 words. No, it's amazing. I get the general premise of being able to, well, like you say, instead of having the side effects being a negative, the side effects are a positive. And doing double duty, having the the program to be able to run those unlimited calculations to spit out things that you never would have thought of and say, oh geez, that's exciting. And
yes.
The amount of money that, like you said, goes into pharmaceuticals could then go into other things, be spread out. So that's great.
Yeah. So for us, We hope this contributes in a lot of ways, but we kind of see the future of medicine. It needs to be accessible, it needs to be affordable, and it needs to work. So I think with this approach, we can accomplish those for a disease that's exceedingly expensive. The best treatment's highly inaccessible at the present time.
Right.
So yeah, we think we're on our way to something hopefully significant.
That's great. That's amazing. Thank you very much for coming in and talking to it. I really appreciate it. And hopefully we get a chance to talk again when a little bigger. Maybe your helicopter can fly in on the roof then once you get it all figured out. That would be awesome. All right.
Thank you.
Thank you very much. Andy Roberts with Teos Pharmaceuticals with spellbinding program coming up that could change a lot of lives globally. Thanks very much, Eric Jingles for BuzzLocal.tv, and we'll see you again. Thanks, Andy.
Hey, no problem.